Good morning. I'm Michael Dong of MWD Consulting. Today, my topic is an overview of the key differences between small molecule drugs and also biotherapeutics. Here's my full agenda for the webinar today, starting with introduction and perspective of some definition and some industrial and business perspectives. The second one is the key differences between small molecule drugs and biopharmaceutical using monoclonal antibody assay model. We cover differences between the drug substance, drug products, pharmacological property and regulatory perspective, and ending up with quality attributes. Thirdly, we would give an example of the case study comparison of two different classes of drugs, of Tarcevo, small molecule, and Herceptin for monoclonal antibody. In this slide, we define what is a drug. According to the Federal Food, Drug, and Cosmetic Act, FD&C, a drug is a substance intended for use in the diagnosis, cure, mitigation, treatment, or prevention of a disease for use as either a component of a medicine, but is not a device. So by that definition, a vaccine is a drug because it's used for prevention, while a cardiac stent or a prosthesis is not a drug, but are medical devices. Some definition, drug substance, DS, also synonymous with active pharmaceutical ingredient or API, can be a synthetic small molecule drug or a large biomolecules, can be a single component or a combo or mixture of therapeutic properties. Drug products are actual dosages being administered to the patients, also known as formulation, could generally tablet, capsule, or parenterals. Also, the term new chemical entity is synonymous with a new molecular entity or ME. This slide summarizes the top 10 drug product by sales in 2024. The actual sales are noted here in small numbers. Note that in the top 10 drugs, 5 of them are monoclonal antibodies, so definitely is a mainstay therapeutic modality. 2 of them are peptides, which are considered a small molecule, and 3 of them are small molecule drugs, with 2 of them are actual combo or combinational product. So those are the lineup on 2024. This table summarizes the key differences of typical small molecule drugs versus biotherapeutic using monoclonal antibody as a model compound. For drug substances, small molecule drugs are solid, either crystalline or amorphous, while monoclonal antibody are lyophilized powder as a drug substance. General property, small molecule, a single high purity, stable organic compound, while biologics are complex, heterogeneous biomolecule produced generally by recombinant DNA biotechnology. Molecular weight for small molecule drugs range from 300 to 700 AMU, while monoclonal antibody typically is immunoglobulins with 150 kilodaltons. Manufacturing for small molecule drugs are by organic synthesis, while monoclonal antibody is performed by fermentation and bioprocessing. The drug product are mostly for small molecule, a tablet or capsule for orally bioavailable dosage forms. For biologic, mainly parenteral, IV or subcutaneous injection because it's not orally available, will not survive the stomach conditions. Storage at room temperature for small molecules and refrigerated for biologics. The key differences of these two categories in terms of pharmacological property, regulatory, and quality attribute are summarized in this table. Mode of action for small molecule drug, typically it binds either an antagonist or agonist, binds with the enzyme receptor or intracellular target, and is generally less targeted, while monoclonal antibody binds to specific antigens with a target which is extracellular target and is very specific. The pharmacokinetic or PK is hours for small molecules and days or weeks for biologics. The dose is based on maximum tolerated dose, or MTD, for small molecules, while for biologic, the dose is based on efficacy determined by a biomarker. The regulatory basis, one typically says that small molecule drug, the specification defines the product, while the actual process, the bioprocessing or the fermentation processes, defines the product for biologics. The regulatory bindings are similar for IND for starting the clinical trial, and for commercialization, small molecule require NDA or new drug application, while in the United States, it's called BLA, or biological license application for biologics. For generic, bioequivalent studies are done without clinical trial for small molecule drugs, while for biotherapeutic, you require comparative studies, and clinical trials are needed to demonstrate safety and efficacy. For quality attribute, there's safety and efficacy with some details spelled here, and these would be detailed in the second webinar, the follow-up webinars. Here are the case studies to illustrate some of the key differences. We're using two drug products, Tarzivo and Herceptin. Both are from Genentech Roche products. Tarzivo was developed and marketed by Genentech. It was a first-generation EFJET inhibitor used for non-small cell lung cancer. It was approved in 2004. It was a blockbuster drug with sales of close to $2 billion at peak years. Herceptin is actually a blockbuster, one of the first blockbuster monoclonal antibodies, also developed by Genentech for treating HER2 positive breast cancer, approved in 1998, and usher in the age of personal medicine because it required a compendial diagnostic for the patient with HER2 positive patients. Herceptin has enjoyed many years of over $7 billion in global sales at its peak years. The current global sales are less than $2 billion after the introduction of many biosimilars. Here are some figures to illustrate the key differences. You can see that the monoclonal antibody is a much larger molecule, 150 kilodalton, versus Tarzivo, which is much smaller. Schematic of immunoglobulin. These are Y-shaped immunoglobulins, such as IgG, 150 kilodaltons, with 1,300 amino acid residues. There's two sets of identical heavy chains shown here, and two sets of identical light chains shown here. The FC region are heavily glycosylated, and these are the FC region, and there are 12 interchain disulfide bonds. This table is illustrated the case study of the differences of drug substance and drug product of Tarziva versus Herceptin. In terms of drug substance, the forms for Tarziva is white powder, crystalline, a phyllolized powder for Herceptin. It's a single high purity stable compound for Tarziva, complex heterogeneous biomolecule produced by recombinant DNA technology, and is definitely much less stable. Molecular weight is 429 for Tarziva and 150 kilodalton immunoglobulin for Herceptin. Manufacturing processes, organic synthesis, purification by recrystallization to yield the stable polymorph. While monoclonal antibodies are generally produced by fermentation using choke or Chinese hamster ovarian cells grown in a bioreactor and purified by chromatography. And once again, these would be detailed in the later slides. Dosage form is 2500, 150 milligram film-coated round tablet taken once a day for Tarziva, while the API is a white phyllolized powder with excipient, which are buffer, and also polysulfate surfactant and sucrose. These are reconstituted for IV administration using the sterile water. Dosing is every 1 to 3 weeks. Storage for Tarziva is shelf life of 3 years, room temperature storage, refrigerated for monoclonal antibody Herceptin. Here is the key differences on pharmacological property, dosing regimens, and regulatory and generic introductions. So mode of action is an inhibitor for EGFR for non-small cell lung cancer. Herceptin binds to overexpress HER2 positive receptor overexpressed in some patients with aggressive metastatic breast cancer, and it would slow the growth of these breast cancer in these patients. Pharmacokinetic is hours for Tarziva, while days and weeks for monoclonal antibody. The dosage is taken once daily, while Herceptin is dosed intravenously with the median weekly or every 3 weeks. Regulatory filing and generics approvals for Tarziva was the first generic was approved in 2015, while the comparative studies and clinical trial were performed to demonstrate safety and efficacy for any biosimilars for biologics. So here's the summary of the webinar. This webinar provides a primer for scientists in pharma and related industry on the key differences of small molecule drugs versus biologics in terms of physicochemical and pharmacological property, and also some drug quality attribute. The webinar allows a better understanding on how to translate some small molecule regulatory and developmental and analytical chemistry experiences and practices into the biopharmaceutical arena, and the detail will be provided in webinar number 2 on the CMC differences. Finally, the references to book chapters shown in 1 and 2 with the book shown here, and some of the references for future referrals. On that note, I'd like to thank you for your attendance and see you next time on the CMC differences between small molecule and biologics.